All right, everybody, I'm going to go ahead and get started. Well, I guess this is probably, I was guessing around 25% of the class today. So I think we're about there. Okay. So, which is to be expected. We're never more than half, except have a quiz or something. All right. So, it's time to move on. Now, I want to talk to you about cancer today. Let me preference this lecture in saying we could have a whole course on cancer. Okay, there's been a lot of advances in the last 20 years, especially in regards to treatment modalities. I'm not going to go into that. What I want to do is to give you a broad overview, very high level, what cancer is, what it is not. And that's about all I'm going to have time for. All right. Now, typically this lecture is, well, because you guys are coming in person, the lecture probably, if I'm lucky, will take about an hour. And I guess you'll get that extra 20 minutes or so. Okay. So it's not going to be the full lecture period for this. But anyway, it's fine. Time for questions if you wish. All right. So before we get into cancer, I want to go over what it is. Okay. So remember, you began life as a fertilized ovum, a single cell. Then you start to undergo mitosis, and then there is the initial development of various stem cell populations. They then controllably differentiate, and ultimately you. Okay. Now, once you're grown and everyone here has stopped growing, you maintain your body form. That means that various cells and tissues in the bodies have a defined lifespan, they die, and they need to be replaced. This is subject to adult stem cell populations that reside in your body. So basically, it is this balance of cell removal, cell replacement that ongoes throughout your entire life. Right now, these stem cells, as I've mentioned, are laid down early in embryonic development, and they then can, to a certain extent, they go from stem cells to progenitor cells to final differentiated states. Right now, and this, when everything works, everything's fine. But let's talk about cancer. Cancer is when we lose this balance. So first off, what is cancer? Cancer is a population of abnormal cells that occur within your body. And basically, they have a propensity to outgrow their normal neighbors. Now, let me really preference this. That doesn't mean that they necessarily have a fest or mitotic rate, a rate of cellular division creating daughter cells. It's that they just don't stop. Some cancers can actually grow at the rate of growth. Their mitotic rate is very slow, but they just don't stop. So over time, they can overpopulate a given microenvironment. All right. Now, these resulting cells will proliferate in this site of origin, which we'll talk about how that comes from in a moment. And they are retained within this site. Eventually, what can happen is they can then undergo additional changes where they can become mastitic, which means they have the ability to escape their site of origin. Now, the first thing is cancer as a disease actually represents hundreds of different diseases. And that's the nature at which it occurs. Where does cancer come from? It represents somatic mutation. What do I mean? I'm talking about when you have two cells undergoing mitosis. This involves doubling of the DNA, splitting it to two daughter cells. There is subject to mistakes. Mistakes do occur. All right. However, somatic mutation means that it does not incur your germ cells. That means you can't pass it on to offspring. Also, it means it is not contagious. Cancer is non-contagious. There is 3 exceptions, which I'll talk about at the end of this lecture series. But for humans, no. Okay? So that means that, let's say you have 75 trillion cells. At any point in time, maybe 1 or less than 1% of them are undergoing mitotic division. That means they're subject to mistakes. Now, without going into too much detail, the cellular machinery in your body is designed to take into consideration and correct those mistakes. It recognizes that there's a mistake. This is going to be an abnormal cell. Let's stop this mitotic division. Let's then destroy this aberrant cell. However, if it escapes the normal policing that happens, it can result in this abnormal cell. And this represents a somatic mutation. Now, the thing to remember, these somatic mutations are completely random. I repeat, they're completely random. That means that any of these genes can become aberrant. Now, does that necessarily mean, for instance, let's say that the mutation is a single amino acid. Number one, it depends on what that amino acid is. It depends on what protein it's associated with, where is it at along that amino acid chain. Most of the time, it doesn't matter. But there can be a particular instance where it can change the functionality of that particular protein. Is this important? Maybe, maybe not. It all depends on where you're at. And we'll discuss certain aspects where, even though they can be completely random, if there are certain parts of a cell function that are more subject to changes. Okay, and we'll talk about, and most of these are related to intercommunication, which we'll talk about the middle of this talk. Now, so even everything's random. Any place you have mitotic cells, you have the possibility of somatic mutations. You have the possibility of resulting cancer. So basically, since it's random, you never know where it's going to happen. You never know which proteins, which cellular processes are going to be screwed up. It has to represent hundreds of different diseases. Now, because let's say though, that it's associated with muscle, it's associated with nerves and blood, etc. Because it associates there, and those cells were originally from that tissue, that means it shares some similarities with the tissue of which it was derived. Right? We'll talk about a little bit of that later on. Right? So, a couple terms. Tumor is a mass of cancer cells. All right? Tumors, there's two types of tumors. So initially, when you have that mutation, it develops into these abnormal cells that gain a innate growth potential over their normal counterparts. They start to undergo hyperplasia. We'll call this a benign tumor. It resides in its site of origin. It cannot leave its site of origin. It cannot kill you. The thing to also remember, I'll interject this right now, benign tumors, benign cancer does not kill you, with one exception. Anybody, what do you think? Where do you think a tumor of any kind can be deadly? Exactly. So a benign tumor is an abnormal outgrowth. Anywhere else, you don't get a pressure buildup, but in the cranium, you only have a limited volume. So even a benign tumor is deadly, has the potential to be deadly. All right?
To have a benign tumor.
Well, all tumors are going to start as benign. So if you're talking about a glial tumor in the brain, what are the statistics? Toward the end, I'll show you some statistics. They're fairly rare. Thank goodness they're very rare, but they can happen. Okay. All right. So enough of the introductions. And then benign means that it's residing. Then it undergoes these additional mutations, and now it becomes malignant, which means it can escape its site of origin. And it can representative cancer cells can travel to other parts of the body where they set up colonies, and this is typically what kills you. For instance, guys, you live old and long enough, you're going to have something called prostate cancer. Prostate is a reproductive accessory organ, which means it's stimulated by testosterone. Now, it is basically, here's your bladder. At the base of your bladder where the ureter is exiting, the prostate surrounds that, okay? So if it is a benign tumor, who cares? All right. In fact, as you get an old fart like me, your prostate it enlarges. That means you have, there's issues. Okay. Now, this has nothing to do with having prostate cancer, but if you get prostate cancer, most of the time it grows slow, but it doesn't kill you. It's when it metastasizes that it represents deadly aspect, because where is it going to go? It's going to enter the bloodstream. It can go to your liver, and if it destroys your liver, you're dead. It can go to your bones. It can go to your lungs. So basically, it's the metastasis in most cancers that kill you, with the exception of brain tumors. All right. Now, this should be obvious. Since cancer represents somatic mutation, that means it's genetically associated. So it begins with these screwed up DNA and these screwed up resulting cells, these aberrant cells. Right? And all of the behavior, benign as well as malignant, is subject to these alterations in these cells. So it's all genetically associated. I told you that all cancer is a result of somatic mutations, that you can't pass it on. However, there are something called familial cancers. What are they? Again, you're not born with cancer. However, you can be born with an increased risk of a resulting somatic mutation. Let me explain this. All right. As I mentioned earlier, within your cell, there is extensive machinery that is designed to recognize and correct mistakes as a result of mitosis. All right. Now, some of these processes are related to certain genes. Let's talk about one, retinoblastoma gene. It is one of these policing genes. So it recognizes these aberrant cells and it tells the cell not to continue on. And it stops this mutant cell from occurring. All right. So in one type of familial cancer, you can have a non-functional RB gene. So it goes by traditional Mendelian genetics. You have 2 alleles per gene. And basically, you need to have a double recessive, which means that you don't have a functional RB gene. So you can have 2 parents. If one of them has both genes, both functional alleles, the other one has one functional allele, one not, they come together, there's the odds 25% that you're going to have somebody will be a carrier. Because remember, you have to have both knocked out. But if you have 2 parents, one with functional, non-functional, functional, non-functional, they combine, there's a 25% chance that I have a child that has a double recessive, which means they don't have a functional RB gene. It does not mean you are born with cancer. That means that this policing situation doesn't work. So somatic mutations occur no matter what, but you have lost the ability. One of your primary corrective measures is now gone because of this double recessive, this loss of functional RB gene. So that means shortly after birth, because remember, think about that, there's a major growth occurring post-birth. All that mitosis. So there's going to be mistakes, but they can escape. And usually what can happen, these retinoblastoma genes variants, these kids can get cancer within 6 months of birth. It also tends to target your eyes. And so blindness is another issue that can come out of that. But that's what is a type of familial cancer. But it's cancer risk. I repeat, it's cancer risk. You do not inherit cancer. Okay. Now, as I mentioned before, it's a random process. However, there are certain genes which tend to be more sensitive. In other words, if I screw them up, they're more likely to result in the possibility of a resulting cancer. And we call these oncogenes. And basically, these are genes that are screwed up due to these somatic mutations. And I'll give you some examples in a bit. Okay. Also, I want you to be clear, is not a single somatic mutation does a cancer make. There has to be multiple mistakes. All right. And we also have to have there's an additional mutation event between a benign and a malignant phenotype. So cancer is a multi-step process. Okay, what you're looking at here is not multiple different types of cancer in the single person. This is time. Across the x-axis here, we have time. So we come up here, we have a single mutation, somatic mutation. This creates an aberrant cell. And let's say that this mutation is going to result in a growth benefit over its native control. So it's going to undergo hyperplasia, right? Otherwise, it's a functional cell, so it's just simple hyperplasia. There's another event, somatic mutation, and now we have dysplasia, which means that this isn't, even though we're getting these multiple pocket of cells, they're non-functional compared to the normal counterpart. So we call it dysplasia. All right. So eventually we get this in situ cancer. It's benign because it resides in the site of origin. There is an additional mutation event. Now we transition from benign to malignant. Malignant means that the ability, these cancer cells have the ability to leave their site of origin. They can then enter their bloodstream, travel to wherever in the body, set up shop, developing colonies. Okay. And this is typically what will result in death eventually. Now, I want to use the next two slides to introduce a concept called cancer stem cells. This was first proposed back in the turn of the last century, the early 1900s. And there was this hypothesis that cancer was a result of a stem cell population that then developed cancer. It was poo-pooed in subsequent decades and just kind of went to the dustbins of science. But in the last 20 years or so, it's gained a huge resurgence. We now recognize, since obviously we know a lot more science than we did in over 100 years ago, and we now appreciate the significance of cancer stem cells. All right. So first off, where do they come from? They can come from a variety of things. Remember, so long as you have a dividing cell, they're subject for a somatic mutation. Right? It doesn't matter. So we can have potentially environmental factors and or just simply luck, bad luck, and you get a mutation. So you have your stem cell niche. And so one of the things about stem cells is they self-replicate and maintain their population throughout your whole lifetime. All right? And that's fine. So they're always undergoing some level of mitosis at some point in time. Now, what could happen is there could be a mutation that occurs. So now we have these stem cells, but they are now cancer stem cells because of these mutations. That's one level. Another one, we can get the next level down. We have stem cells, progenitor cells. They are still mitotic. They can have a mutation because they can undergo mitosis. These can then develop into these cancer stem cells. Now, the progenitor cells themselves then become differentiated cells. So long as those differentiated cells still have the capacity to undergo mitosis, there is also the possibility that they can get somatic mutation, which could also potentially result in these cancer stem cells. Now, these cancer stem cells have the capacity to completely regenerate a tumor. In other words, if I knock out all the cells in a tumor except the cancer stem cell, it has the ability to recapitulate that tumor. All right. So the reason why I'm talking about this is because how we treat tumors. How do we treat cancer? So at one time, we just wanted to knock out the whole tumor. We tried to knock out the whole tumor, and most of the early chemotherapies, it's a toxin, it's a poison, and you're trying to come up with a poison that will preferentially kill the cancer cells, but not kill the normal cells, all right? Those drugs have evolved over the decades. We still have a long way to go. But what I want to say is that what's more important than trying to knock out all the cells, what you want to knock out is that stem cell population, that cancer stem cell population, because if you have these tumor cells, that means that, again, this is how we're getting them here. Remember, if we have mature cells that are post-mitotic, they can't become, there's no potential for them to become cancer cells because they're not undergoing mitosis. So they're not subject to events. But we have a tumor population. It's heterologous because it's got all, yes.
Previous slide, what did that lightning bolt signify before this? Like some sort of stimulus?
What are we talking about? This
That
environmental
lightning.
factors here?
No, that lightning bolt in all this sense. The yellow one.
Oh, this here. That just means that's the somatic mutation. That's where the mutation occurs.
Some stimulus of.
Well, no. That's where they're undergoing mitosis, and now you have a resulting somatic mutation. That's what the little lightning bolt is. And the second slide is basically the same thing I was just talking about. Okay? Right? Okay. Now, the reason these cancer stem cells are important because there's a lot of new drugs that are being developed that specifically target the stem cell populations. Why is this important? Remember, a tumor is going to be heterologous. There's all kinds of cells, and not all of them are cancer cells, and some of them include cancer stem cells. So if we come over here and we give a chemotherapy, it knocks out all of the cells with the exception of a cancer stem cell. That means that resulting surviving cancer stem cell can completely regenerate that tumor. All right. However, if you knock out the cancer stem cell population, the remaining cells will eventually die and degrade, and so you will lose the tumor. So right now, a lot of these chemotherapy drugs are being developed to target the actual cancer stem cell population, subpopulation. All right? Okay. Let me talk a little bit about, characteristics of this mass of cancer cells called tumors. All right? Always remember, cancer tumors are derived from normal cells undergoing somatic mutations. Also remember that these resulting mutant cells have a aberrant growth behavior. They outgrow their normal counterparts, but not necessarily growing at a faster mitotic rate. They just don't stop. Also remember that this hyperplasia results in crowded conditions of these resulting cells. So the tumors are developing in crowded conditions. Crowded conditions means less available oxygen for the constituent cells. So those cells are existing in an anaerobic environment versus one of high oxygen. And this has implications down the road. Because they're growing in these crowded conditions, the normal architecture, geometry of that tissue is removed. So they're not subject to neighbor-to-neighbor interactions, and cell shape they're independent of cell shape. So next, though, for that tumor mass to get beyond a millimeter cubed in size, it needs a blood supply. So that growing tumor eventually, in order for it to get bigger, will require blood vessels. Now, the thing about the vasculature of tumors versus natural tissue capillaries, they're quite different. The tumors and the capillaries within tumors are, number one, the vessels are very thin compared to normal capillaries in normal tissues. There's also more fenestrations, more holes. Think about Fick's. What this means is the rate of exchange between the plasma compartment, the blood, and the interstitial compartment is greatly facilitated. Okay? This results in the net movement of two things. Water, number one. So that means that the hydrostatic pressure within the tumor is greater than normal cells. The implication of this is in drug delivery, because that means that in order to get a drug from the blood into the tissue compartment of a tumor, we have to overcome this greater pressure, which is resisting the outward movement eventually. Number two, it also means this higher transport means that oxygen, nutrients, all of these things are preferentially going to the tumor versus their normal counterparts. Right? All right, now, again, let me reemphasize that we can't, any place where you have mitotic cells, you can have somatic mutations. So here is the normal pathway, which I talked about in the beginning. We have a stem cell population. It replicates itself. Some of those are destined to become progenitor cells, and those then become terminally differentiated into whatever mature cells. All right. And they don't stop dividing. Right? Over here, what if we have the mutation occurring in the stem cell population? They never differentiate. So they're always this mass of stem cells, but they don't differentiate. Means they're not making whatever terminal cells, mature cells they need to. Now, you could also have resulting daughter cells anytime during mitotic phase being screwed up as well. Okay. The next concept I want to talk to you about is tumors being either polyclonal or monoclonal. What do I mean by that? In the case of polyclonal tumor, that means you have simultaneous somatic mutations occurring within a tissue compartment. That means multiple cells undergoing mitosis are having simultaneous mistakes made. So in other words, you're having multiple aspects, you're having all the mutations occurring at the same time. All right. This does not happen for the most part, well, essentially does not happen in humans. Humans are referred to as monoclonal tumors because they are essentially most tumors, if not all tumors, are derived by a single mutation within a tissue compartment. And then there's, over time, additional mutations within that mutated tissue. That ultimately results in tissue. Okay? A tumor, excuse me. Now, so let's kind of go over a summary of the process of tumor formation. So we have our normal condition, which means I have stem cells, we get progenitor cells, we get post-differentiated mature cells, and we maintain a tissue. Now, what happens during the mitosis event? We get a somatic mutation. We get a abnormal growth potential for that resulting mutant cell population. It replicates, creates more daughter cells in comparison to its normal counterparts, creates hyperplasia. There's an additional transformation event, and we now lead to in situ cancer, a benign tumor. All right. Then what happens is somewhere along the way, we have an additional somatic mutation, additional transformation. And when I say transformation event, I mean a mutation. Okay. So now we have an additional mutation, and now we switch from benign to malignant. And I'll explain the difference here momentarily. All right. So basically, it's a multiple hit process that we have to go through. So you just don't get that to begin with. And let me use this little simple example to illustrate it. At the top, we'll see a normal cell population. They're all white circles. There is somatic mutation event 1, transformation event 1, and that represents that quarter black pie. Okay. Notice it happens in a single cell. Then we have a preferential mitotic division of that mutant cell population. So we get now have a tissue compartment, keeping it simple, 2 cell types. One's normal, and one of them has a single mutation. Right? Now, at some point, this mixture of cells, one of those quarter pi mutant cells undergoes an additional mutation, and now is a half pi. All right. So now we have 3 cell types. We have the normal cells, we have the quarter pi, and the half pi. Now, and all of those are dividing. Then one of those half pies undergoes an additional mutation. Now it's 3/4 pi. So now we have this heterologous tumor, which contains normal cells and cells all along that somatic mutation cascade. All right. So that is this. I really want to reemphasize here that cancer is a multi-stage process representing multiple in time subsequent mutations within that initial mutated group. Yes. I want to say no. However, it depends on what that mutation is. That potential mutation, for instance, if that mutation was to hit my RB gene or something like that, that could lead me to the propensity to have an additional mutation. But generally speaking, no. Okay. And then we'll focus on in a minute about oncogenes, because what that means. Okay. Next, remember these mutation events are random. They occur in any dividing tissue, and we can use where's the site of origin of a tumor is basically we can use that for nomenclature purposes. So basically, carcinoma refers to any type of cancer that results from somatic mutations occurring in epithelial cells, right? So breast cancer, ladies, those are mammary epithelial cells, so that would be called a carcinoma. You can also have a sarcoma, which includes musculoskeletal tissue. Maybe that's muscle, maybe that's bone, okay? You also have leukemias, which means cancers associated with blood cells. Usually these are white blood cells like monocytes. Or lastly, you can have gliomas, which involve nerve tissues. Okay, so those are the 4 basic classifications of cancer. It all depends on what source. What was your tissue source? Next, we can divide them into, are they benign or are they malignant? Okay, I'll just give you one example. We'll talk about an epithelial tumor. So let's say you have breast cancer. It is a carcinoma. It would be an adenoma, which means it's benign. As it then transitions to malignant, now it is an adenocarcinoma. So nomenclature can actually tell you where it's from, what tissue it was derived from, what cell type, as well as whether it's benign or malignant. Okay, let's focus a little bit more on what differentiates benign versus malignant, other than additional mutations. So on the left, you'll see a benign tumor. This is the primary tumor. It resides in its source of tissue of origin. We have this local overproduction of cells. Eventually, it will put stress on the surrounding normal tissue. The body will try to contain it, and so it will surround it with a fibrous capsule, and now you will have this benign tumor. But it cannot escape its site of origin until there's additional mutations. With additional mutations, it gains the ability to escape. What this primarily consists of is most malignant tumors, or essentially all of them, have one of the mutation results in exaggerated protease production. Why is this important? Of course, in order to escape this fibrous capsule, which is defining this benign tumor, the malignant cells now degrade this capsule, which means the cells can spill out into that environment. Because of these proteases, that facilitates migration, and the cells migrate to a vascular supply, whether it is the lymphatics or the blood. They can then enter those fluid compartments and then be transported throughout the body. Okay, so benign, malignant, contained, non-contained.
So that's what that is.
So when, let's say that they're excising a tumor and a benign tumor, basically what they are always looking for is the border as well. They want to make sure that you want to exceed this border region. You don't want to leave the possibility of a cell out, cutting it off here and leaving the tumor, some of the tumor cells behind, right? All right, at this point in time, and I'll circle back to that. Before I do that, I want the reason why I pop cancer yes.
This slide,
Mhm.
it shows like that capsule of like fibrous connective tissue on the benign side. Is
Yes.
that more so just like how the cancer grows, or is that like kind of?
No, that's the normal host tissues trying to they can't do away with it because they don't recognize for whatever, and we'll talk about different strategies. It doesn't recognize it as a foreign body that you have to remove. I can't remove it, I want to contain it. So the thing to do is that you surround it with this fibrous structure to try to keep it in place. And that does hold it in place until we have an additional mutation. Yes.
You said protease production? What's
Protease. It's an enzyme that destroys proteins. Okay. All right. Moving on. I put this discussion following endocrinology. That's because what are we talking about? We're talking about mutations that result in abnormal cell growth. What controls cell growth? Hormones. Okay. So the thing to remember is that a lot of cancers are a result of mutations associated with some aspect of endocrinology. Now, I do not say that if I give a drug, I'm going to induce a cancer. What I mean is if I give growth factors or hormones that really promote mitosis, I'm going to increase the chances of a mistake, right? Now, the other thing is that cancers represent screwed up genes, all right, or these mutations, all right? Now, what happens if so what we want is those screwed up genes we call oncogenes. Most of these oncogenes are associated with endocrine communication, all right? Now, as I'll talk about in a moment, some of those mutations may result in an overproduction of a particular hormone called a growth factor, which means that the cells are those resultant mutant cells grow at a faster rate, all right? Now, another way at which hormones and cancer are involved is depending on the cancer, sometimes we can treat cancer or diagnose cancer via hormone receptors or hormones themselves. Guys, I mentioned prostate cancer. Okay, prostate cancer, the prostate is a male reproductive organ, accessory organ. It is responsive to testosterone. So one way at which I can control the development of that tumor is to reduce testosterone, right? Now, no, we do not result in castration, but we can give drugs that will suppress testosterone production. Reduced testosterone production means a reduced growth rate of that developing prostate cancer, okay? And that is used. Second, anti-hormones. And so that's basically an anti-hormone. When receptors, ladies, breast cancer, one way that we delineate breast cancer is whether there are specific types of receptors within that cancer. Because if they are there, if there's mutations do occur there, we will treat that differently. Treatment is completely different than where they don't have those receptors. So that can influence treatment. So there really is a very close association of the endocrine system and cancer, simply because a lot of the genes that are screwed up will impact cell-to-cell communication. Okay. Now, what does that mean? What can be those mutations event? How does this affect the endocrine communication cascade? Number one, autocrine secretion of a growth factor / hormone. So back in 1978, there was a hormone discovered, a growth factor called transforming growth factor 1. As the name implies, it was thought that this induced cancer. Because if you treated cells with this, they lost their ability, that they gain the ability to grow in contact-free situations, which at the time was a definition of a cancer cell. So the name was transforming growth factor beta. Within 6 months, they were finding this in normal tissues. So that lost its effect. But what happens is this, one of those mutations were that you get the overproduction of TGF beta. TGF beta gives an overproduction. It creates an immune regulatory environment, which means that the immune system is less likely to attach it. It creates a more amiable environment for the creation of a cancer. All right. Now, this is autocrine secretion because the cancer cell is secreting, it acts on that cell, and it gives it growth advantages compared to its normal counterparts. Number two, you can maybe independent of overproduction of a growth factor, we have overproduction of a receptor population, the number of receptors per cell. So that means, as we've already talked about endocrinology, that gives a certain growth potential for those types of cells because they're more sensitive to a given level of a hormone. Next, loss of growth inhibitions. So what can be associated with this is even with normal growth factors in the surrounding environment, those cells just don't stop growing. They just continue to grow. There could also be, has nothing to do with the hormone. It has nothing to do with the surface receptor, but what about the downstream cascade of effects that occur inside the cell? They can get screwed up by mutations, which means that could result in, even though I'm getting one hormone to receptor interaction, but instead of resulting in that action resulting in 10 million events, what if it resulted in 100,000, 1,000 million events, a billion events, right? And lastly, what also happens, especially during metastasis, is we have the overproduction of these proteases. They do not only increase the capability or potential for migration, but they also will increase the availability of available resources, i.e. growth hormone, various hormones and growth factors, and they have better stimulation of those cells. Okay, so all of these can be a result in abnormal growth. All right. And now I said oncogenes. And most of a good chunk of these oncogenes are associated with endocrinology. All right. So some of these oncogenes can actually result in an overproduction of a growth factor. An example here, platelet-derived growth factor. So what happens is that oncogene results in overexpression, autocrine secretion of PDGF. It can also be KRAS or MDM2. Those are some of those policing genes. If it knocks those out, that means it doesn't affect mutation rate, but increases the chances of a potential survival of a mutated cell. They can also have nothing to do with the growth factors, maybe, and these are also these tumor suppressor genes, which I talked about. RB, which I talked about, and there's BRCA2, BRCA1, and NF1 as well, because again, this is the ability for randomly occurring mutations to survive. So growth factors, oncogenes in cancer, so they can be growth factors themselves. The aberrant genes could be receptors, or they could be the signaling pathways themselves. Let's skip. Well, actually, let me go back to this one. Okay, so this is a cell cycle, right? So when cells undergo their division cycle, you have a cell which is in an MO phase. That means it's non-dividing. Now what happens is we have to proceed to a G1 phase. And a G1 phase is where we're going to get to prepping in order to have mitosis. And that means to have duplication of the DNA, and then the DNA separates, right? Now, what you're looking about here, all during this cycle, remember those, there are genes, we have the police genes, which are going to influence. There's a mutation, let's don't continue, let's don't divide, and they work. So that RB gene works here. If we say stop, we don't undergo mitosis, we don't then replicate that cell. All right. So this is just a normal curve. So even though, and we only have the possibility of mutation during DNA synthesis or mitosis. Now, if you remember in the introduction series, I told you that the immune system is designed to do two things: to protect you from outside invaders, as well as aberrant cells in your body. All right, these mutant cells, which can become cancer, are aberrant cells. However, of course, the obvious question is, why does the immune system take care of it? Because these have gained the potential to escape detection or elimination by the immune system. All right. How does that work? What are the immune changes associated with tumor development? Number one, it could be that these aberrant cells are not producing antigens that are recognized as different. So in other words, the immune system doesn't recognize it as non-self. It's not different enough to be recognized. If it's not recognized, it can't be eliminated. Number two, there is also homogeneity of antigen suppression by tumors. So what do I mean? That means that maybe what's happening is the tumor it's forcibly not expressing these immune identifiable proteins, the antigens. All right. Now, what also can happen is that there are on these cancer cells, but when immune cell is going to is trying to monitoring the system and sees them, they can release those antigens from the surface. Now, if they're no longer associated on the surface, the immune system doesn't see it as an enemy. Okay. These can be either by internalization or release and secretion into the interstitial space. Lastly, which we are really understanding in the last 15 years, even more importantly, is that these cancer cells are immunoregulatory. So in other words, they suppress the immune system such that it does not attack those cancer cells. But this tends to be associated with the site of the tumor, but it can also gain to be systemic in effect. But these are all possibilities whereby developing tumors escape immune detection and continue to grow. All right. So, metastasis. We have a benign tumor. It now is going to become a mastitic tumor, which means it can escape. What does this process entail? So basically, that means that, as I've already shown you, you have your primary tumor, your benign tumor. It's contained in a fibrous capsule. Now we have converted to a mastitic tumor. It has the ability to escape. The cells pour out into this normal tissue compartment. They migrate, they enter a vasculature. Let's focus on blood here. And they can then either via the blood or lymphatics, they can go wherever they want in the body. Typically, that means whatever's downstream. So where the source of the tumor is, if we now have cells being released from that, if they go into the vasculature, then they go. So if they are leaving here, they'd have to go to my foot before they come back up here, right? By the vasculature. And then, of course, if they're going to lymphatics, they're going to have to follow the lymphatic tree and then ultimately get returned to the vasculature. Right? Now, they're in the blood. They're going throughout the body. Then, in order for them to develop successful metastases, they have to leave the blood compartment. So that means they have to squeeze between those endothelial cells, which define a vasculature, a capillary, and then they can leave that into the interstitial compartment where they can set up shop. They start to undergo mitosis and they develop a new tumor site, right? So, and these then is what typically kills you because they can be set up shop in the lungs and the liver, etc. These more organs that, if they're compromised, they can lead to death. Right? Now, a lot of times, and I'll just interject it here because I've got some time for today. So you can have at one time, I used to work I was in orthopedics for 10 years. And one of the first thing I worked on when I first started was osteosarcoma, bone cancer. And in bone cancer, what can happen is you have your primary site. Now it undergoes it's mastitic. Now you have your original source can be say here in the bone. Then what happens? It's releasing mets. We'll call it short for metastatic cells. They go via the vascular. They'll go up, say, here to the liver, or maybe the lungs. Right now, that means at any time post mastitis, I can never say it. So metastasis, we can have multiple tumors. We can have our primary mastitic tumor, say here, and we can have these colonies. Now, one of the things that's interesting, at least in osteosarcoma, is the parent tumor controls the mets. So in other words, it can suppress the mets. So when you go into the 80s, into the early 90s, typically the way you treated osteosarcoma usually would be associated in the leg was amputation. So you remove the tumor, and usually it's associated with the bone, and the only way to do that is to remove the whole leg. Okay? Now, so what you do is initially you would do a diagnostic and say, where's the tumor? You say it's here. You don't see the mets because they're so small, because they're being suppressed by the parent tumor. Now you've done your primary surgery, you removed this primary source. Then what happens over time, the mets have lost their suppression, and now they really start to grow. So the thing about it is, within osteosarcoma, you always had to follow that patient for a while because those mets could come up sometime later. You cannot, and you wouldn't see them in the initial diagnosis because they were suppressed. Okay. So when you think about it, this is scary. Okay. You have these cells, they can enter the bloodstream, then go anywhere. They can set up shop in the liver or someplace. And then if they get big enough, you're dead. Right. But there's one saving grace. It's an extremely efficient process, which I'll talk about. This is a graphic rendition of what I just talked about. So they leave their site of origin, they travel via the blood, they then metastasize in a distant site. The reason why it is inefficient. So what do I mean? For all of those mastitic cells entering the bloodstream, within 24 hours, less than 1% survive. Why? Remember where these cells came from. They developed in situ in a very unique environment. Crowded conditions, remember, lower oxygen, and now all of a sudden they are in blood, which is lots of oxygen, lots of nutrients, non-crowded conditions. Oh, importantly, it's a tortuous mechanical environment. Okay? Think about it this way. Analogy. You're in Lake Erie, okay? And you're leaving Lake Erie, entering Niagara River. Everything's fine. Then you go over the falls. You don't survive. It's all part of the river. It's just that it's mechanically such that you hit on the bottom of the rocks and everything turned up into fish bait, okay? So it's the same thing. So within the tissue compartment, that's where they evolved. It's very static forces. There's no forces. Now they enter the bloodstream and they're just being hurled along, banging across along the sides of the vessels, and they get destroyed. Okay, but you only need one, especially if we're talking about a cancer stem cell to survive, to set up shop. Yes.
Wouldn't cancer stem cells be less likely to survive that environment? Stem cells are more sensitive to their...
That's part of it. So that's why less than 1%. But obviously, it only needs one to survive. Okay, statistics. These are a little old, but they're still relevant. The take-home messages that haven't changed. Guys, prostate cancer. Ladies, breast cancer. Okay, everything else is a little bit less. Lung associated cancers, those keep going down because we are less smoking. However, they're going up because of aerial pollution aspects. Okay, but they are a lot less than they used to be. We could bring in colon and rectum. There's been a steady increase, especially in young people your age. So that's been on the upswing in the last 15 years. Let's see. One of them I will mention here is pancreas or ovarian cancer. The reason why they are disproportionately deadly is the delay it is in identification. One of the things over the last 30 years, ever since the war on cancer began, the survivability has really increased because we've increased our diagnosis. Earlier diagnosis means you catch it at earlier stage. You catch it at a benign stage, it's much better than a metastatic stage because it's where it is. You take the tumor out, you're done. If it's metastasis, like I told you for the osteosarcoma, you got to deal with the Mets, right? And they could be in a place where you can't surgically remove them. Now, the case of ovarian cancer or pancreatic cancer is that they're internal, and usually by the time they're identified, they're already at a much later stage. They're metastatic. And that's one of the reasons why. It's not that they're any less deadly than others, it's just the delay in normal identification. Let's just focus on breast cancer. So, you know, your breast screening. And then you have to worry. I mean, I know it's in the news all the time. It was in the news recently about false negatives and things like that. So if you can catch it, but a lot of it has to do with, for instance, if you have denser mammary tissue, it means it's hard to image through. So you're going to get a lot of false negatives under those circumstances. So that's all has to do with fast screening. Another one, guys, prostate cancer. You don't have to worry about it yet, but when you live a few more decades, like for instance, there is a hormone that is produced by your pancreas called PSA, prostate specific antigen. And that is that was really used about 30 years ago as a biomarker. So, but it's still useful. All right. So normally your PSA level, and you guys, your PSA level is non-detectable. You come me, I don't have an issue. Matter of fact, I just had my annual and I just got my blood work done on Saturday. It was less than 0.4. So well away from 5. 5 is the critical thing. If you go 5 or above, that means you're likely to have prostate cancer because enlarging prostate is resulting in the increase in production. The reason that kind of fell to the wayside for a while was when the levels were starting to go up, guys, oh, I've got prostate cancer, I've got to take care of it. But the thing about prostate cancer is usually speaking, it's a very slow growing cancer. So you don't necessarily want to do anything right away. You want to wait till it gets to a particular point because what are your options? Surgery, you don't want prostate surgery because there's you can screw up all. Remember, here's the ureter, here's the bladder, here's the prostate. Taking that out with all of the boundaries without harming the surrounding tissue is hard. Okay, a lot of times you can end up incontinent. Brachytherapy, that's seeding little radioactive pellets in there to destroy the tissue. That can work, but it can work for a while. My dad had that done. He had 10 years of disease-free, but then it started up again, and ultimately it did kill him. But it was an issue. I had one of my uncles, he had the surgery, and he ended up being incontinent. And you sat on donuts for about 6 months. So there are other strategies as well. There's cryotherapy, which means freezing it. Then Yoed Rabin in mechanical engineering. I've known him for decades, and that's one of the things he's been very good at doing is doing cryosurgery, and that means freezing it. So you insert these probes and you freeze it because also want to only freeze the tissue you want to freeze and not all the surrounding tissue. So that's a problem. So yeah, it can be an issue. And there are no good options. If you can postpone it as long as possible, do it. All right. Next, I want to talk about the concept of where does it come from? Where does cancer come from? There are two general hypotheses of cancer. One is, well, first off, hereditary. Remember, that's just risk. You don't inherit cancer. We can talk about replicative and environmental. What does that mean? Replicative means just bad luck, because it all means it's just random. When these cells are undergoing mitosis, one of them is a mistake, and it's completely random. That's what's meant by replicative. So, however, the more mitosis a tissue has, the greater the chances of having a problem. Okay? So that's why you're going to see a lot of that, for instance, here. All right? Because in your gastrointestinal tract, those lining epithelial cells are replaced every 5 to 7 days, or 3 to 5 days. Okay? So a lot of mitosis. Environmental means that there's something in the environment which induces these mistakes. Radiation, chemicals, viruses, whatever. Okay? So, remember the herpes virus, okay? And then ladies, you can get immunized for that, because that can result in your huge increase in risk in cancer formation, uterine cancer formation. So you get vaccinated against that, it actually protects you against developing certain types of cancer. All right. So there's always been an argument. If you go to any cancer meeting and you get into a discussion about what is the primary source, replicative or environmental, you'll get people on both sides. Well, recently, there was a definitive study, not 2017, which actually looked at 17 cancer types in 69 countries. I mean, it was an extensive study. And basically, it found that everything was driven primarily by replicative, not by environmental. Now, obviously, there can be exceptions, but most of it just boils down to luck or a lack thereof.
So, earlier I asked,
Yes, we do screen. We don't screen for prostate cancer. We don't have a screen for prostate cancer. We don't have a screen for ovarian cancer. We don't have any markers. We do not have a marker. So we don't have, the only way we can monitor brain is do MRIs, and we can't afford to do that on a routine basis. We have no chemical markers for brain cancer.
How do you know?
You just, well, a lot of point, if they're expressing certain clinical signs, and then that warrants having something. So the point is, without a chemical biomarker, like say PSA for prostate. So what you do is, so if you've got repeated headaches and things like that, that suggests, and you've eliminated everything else, and what's left is XY, particularly brain cancer, then that would warrant the expense of having an MRI. Okay. And so there has to be extenuating circumstances to do that, because it's extremely expensive and a pain in the butt. Okay. So that's kind of it. So a lot of it is luck. A lot of times you're going in for a routine physical or something, or you have something and the physician identifies a lump where there shouldn't be a lump, and that identifies, and so that could be it. Again, it's luck. All right. Okay. I told you at the beginning of this lecture that in humans, cancer is not contagious. I want to talk about 3 examples of where it can be, and I'll focus the first one on this one. The Tasmanian devil. It is a marsupial mammal from the country of Australia. It resides in the state of Tasmania, which is a little island off the southeast coast of Australia. Beautiful island. And the thing, how many of you seen Looney Tunes, classic Looney Tunes, where you have the Tasmanian devil, Tassie and so forth? Crazy thing. All right. Tasmanian devils, they actually was a little off, but you know, they don't eat everything. Number one, they're about the size of a big house cat or a small raccoon. They're just 10 times meaner. Right. So in the island of Tasmania, they serve the same purpose as a possum or a raccoon here in town. So it's not uncommon you go into, if you have your garbage can by your back door or something turns it over in the night, it's a raccoon or a possum trying to get at stuff. In Tasmania, that's the purpose they serve. Now there's some unique situations. Even in all other animals, this is not, it is not contagious, but there's a series of events which are uniquely I want to talk about. First, behavior. The way Tasmanian devils say hello is they try to rip each other's faces off. Okay? They're very aggressive species. So they're always fighting. So, and that's part of the mating rituals too. So that means they've got little claws and they've got teeth. You don't really see them here, but they have sharp little teeth and sharp claws. So their behavior is number one. Number two, not too many decades ago, about 30, 40 years ago, they almost went extinct. There are populations residing in zoos around the world, even on the mainland Australia, but the wild species almost went extinct. That means that the resulting population now in Tasmania is all rather inbred. They're all cousins. Okay, that means they're genetically similar. Number three, there was in the early 1990s, or late 1990s, there was a Tasmanian devil that developed a benign cancer that is a maxillofacial cancer. It's in the jaw. It never has gone beyond benign, but it's still deadly. For instance, brain cancer, a benign tumor can kill you because of pressure. How does a benign maxillofacial tumor kill you? It's in the mouth. It grows to such an extent they can't eat, so they starve to death. Okay? So the benign tumor is deadly. They have an inbred population, and they have an extremely aggressive demeanor. All right. So first identification started up here. Here's the first detection, 1996, northeast end of the island. This is when the first mutation, the first cancer arrived. Now, remember, they're always fighting. So next thing we know is very quickly it starts to spread. So what that means is animals are fighting. They scratch each other. They're actually going to, now remember the tumor is in the mouth. They grab some cancer cells and they basically surgically are placing them between individuals. Have these open cuts and scratches and contusions, and then we have the transfer of these cancer cells because the animals are very closely interrelated. It survives and develops. It takes a while. It takes months for the tumors to grow, for them to starve to death. So there's plenty of time for them to pass it on. And so the wave continued to go, and they were projected that within 20 years they would be extinct in the wild. However, what happened just recently, they found that these animals are now, some subpopulation is surviving because they underwent evolution. One of those in population then developed a ability to resist the development of that tumor progression. And of course, that one is surviving, that one's replicating, and the population of Tasmanian devils is no longer in danger of annihilation. This is the most obvious example that I know of, but it's a unique situation that does not happen anywhere else in the world. Now, of course, the question is this the only animal that has where cancer can be considered contagious. No, two more exceptions. Number one, dogs. Okay, canine transmissible venereal tumor, CTVT. This has been around a long time, and it is it's a very specific, very, very rare cancer, and it's specific for dogs. Lastly, marine bivalves. You know what a bivalve is, right? Clam, a mussel, barnacle. Okay, well, not a barnacle; they're not bivalves. So basically, it's a leukemia type disease, and it's contagious. These are the only three instances that I'm aware of whereby cancer can be considered contagious. Okay, not in humans, right? So with that, that is it for my lecture on cancer. Again, it was not meant to be completely inclusive, just to give you the high points of what cancer is and what cancer isn't. All right. So that's it for today. I'm going to let you guys go a little bit early, and we will start the next system on Thursday. Okay. No, obviously no homework, and so I'll post that on Thursday. Okay. Let me stop this first.